In this video, we will discuss about the future directions to improve CAR-T efficacies by combining the AI-driven approach. So, in this video or in this figure, the authors talks about how they can combine the AI to improve the CAR-T cells design. So, first, the main idea is that the artificial intelligence will be used to screen the data, or they will use data mining and find out which receptors works well for which kind of tumor mutations or the tumor heterogeneity and its correct solutions or correct receptors of the CAR-T designs that can help in diagnosing this tumor heterogeneity. These things can be addressed with the help of artificial intelligence in less duration of time, as they will have lots of data and they will also have the results of different CAR approaches that are used against patients of different kind of heterogeneity, and they will use that informations to design a car. To design a car, one approach is to design a universal car, that is, use these informations to design a universal car that can have the informations about which genes needs to be knocked in and which genes needs to be knocked out, and what is the optimum car that can be used. And one of the example is shown here, uh, that is on the top right side. It is a car, it is a T cells. It has the antigens against the tumors, and they will have some of the genes that are knocked down, that is CD155, some of the receptors that are knocked down, that is CD155, CD52, HLA, KO, as they will help in as they will leads as this expression of these receptors might interact with the tumors, and that leads to the suppression of the tumor activity. And this T cell these things can also be knocked down, that is TCR, T cell receptors knocked down, and or these things can be activated, or track dependence activations of this CAR-T can be used, that is, when the CAR is in the tumor microenvironment, then only this CAR will be this CAR-T cells will be activated and perform its functions. So this will improve the efficacies, and at the same time it will reduce the side effects. The other things can be done is to customize the CAR design based on the patient's tumors heterogeneity, or based on genes that are present in that are mutated in the patients, in a specific patients. So here, since the artificial intelligence has the informations that which combination of CAR could work better in which tumor in a tumor heterogeneity, in a different tumor heterogeneity conditions. So this this informations can be used by artificial intelligence to design a system, or to design a system where they can um, make this where they can use this CRISPR-Cas9 system and use the delivery system either in the form of virus or lipid nanoparticles, and the CRISPR-Cas9 system will be designed against the genes that needs to be knocked in or knocked down based on the information provided by artificial intelligence, and these things then packaged into the virus or lipid nanoparticles that has a specificity to target the CAR-T cells, or we can say that in vivo CAR-T cells generations. And since now it is already since the system is optimized against the uh, individual patients, the chances of side effects are very low. However, these things are still in the initial stage, as targeting the or specificity of targeting the right cells at CAR-T cells is still a challenge. The other things can be done is to use these combinations, uh, instead of this using only the CAR-T cells, a combinatorial targetings, or we can say that one can use the other immune cells such as NK92 cells along with the CAR-T cells, so that they will have a synergetic effect, and that will increase the CAR-T it will increase the immunotherapies efficiencies or immunotherapies efficacies against the anti-tumors much better compared to the case where only CAR-T cells are used. So these are some of the future directions that on which these CAR T cells designs can be moved forward, and which might leads to a better tumor efficacies against not only uh, against the B cells malignancies but also against the solid tumors. As currently the challenges against the solid tumors is that the CAR Ts are not able to reach to the tumor sites, and before that they will get deactivated either by tumor microenvironment or or by the conditions that are present in the tumor microenvironment that leads to this tumor microenvironment provide a physical barrier that prevents these CAR T cells to reach to the antigens. So that's all for this video. If you need to understand more about their future directions in detail, you can go through the reference that has been provided in the description. Thank you for your time.